Okay, so what I'd like to do is try out using some of these really basic definitions in an example problem. It's not that different from some problems that we've looked at in the past. Let's imagine that you're doing one of these large proteomic analyses, sort of a high throughput analysis. And let's say in particular we're looking at a protein-protein interaction analysis that's looking for pairs of proteins that bind to each other. And let's say, as was the original case, these studies were originally done in yeast, which has about 6,000 distinct genes encoding proteins, so about 6,000 different proteins total. And let's say, doing some kind of large screen, we pick up 1,000 positive pairs. In other words, our test result is telling us that about 1,000 of those pairs are indicated to be interacting according to this true hybrid criterion. I'm not going to go into any of the details of exactly how that's done experimentally. The point is not those details, but rather just thinking about how these error rates play out in terms of meaning for how we interpret them in real life. So let's say, based upon this data set of 1,000 protein pairs indicated as interacting using an exciting new methodology, okay, it's time to publish a research paper showing that this method is generating results. And let's say that at that time, lo and behold, somebody else is doing sort of the same thing, and your boss comes to you saying, well, they just published their paper saying that they got, not necessarily exactly 1,000, but something roughly in that neighborhood, number of pairs also as interacting. And a sort of obvious question, your boss might say, well, gee, let's compare our list to their list and see how they compare. Obvious question, do we expect the two sets of results to agree with each other? And when I say agree, I mean that a large fraction of the interactions found in study A are also expected to be found in study B as a basic criterion of quality of the studies. If that's not the case, then there's something wrong with, basically that its results are wrong, for example. Okay? So based upon the basic set of different kinds of error measures that we've just been thinking about, try to bring those into play here to ask yourself whether you would expect this particular situation to require that those two lists of protein-protein interactions, basically that the lists more or less agree with each other, or whether that's not really expected to be required as a basic criterion for the results in those lists to actually be correct. In other words, that they are real protein-protein interactions. If you expect them to need to be in agreement with each other, briefly explain why. If not, briefly explain why not. So if you join the live session, you should see this question now up on your computer. Any questions about what you're being asked here? For example, this criterion of should, exactly what I mean by that? Okay, well, why don't you go ahead and think about that for a minute or two, and then submit a brief answer.
Do we know the 1,000 protein-protein interaction is the only protein-protein interaction in the 6,000 protein pairs?
So he's asking the question whether whether the result of detecting 1,000 protein-protein interactions is guaranteed to be complete. Is that basically what you're asking?
Almost complete.
No.
Okay.
No, there's no such guarantee.
Oh, and there may be like wrong interactions, right? In the 1,000. We don't.
Right.
expect
Just because a result is reported by an experimental method doesn't mean that it's automatically right. Sure. Great question. Why don't you go ahead and finish entering whatever brief answer you come up with.